Namaskar. Welcome to the session on food quality and sensory evaluation. Today's session is about odor measurement techniques. Firstly, introduction. Human olfaction is a protective sense as it allows the detection of odors which helps for food acceptance, recognizing danger and disliking the unpleasant. Odors are a mixture of light and small molecules that coming in contact with various human sensory systems also at very low concentrations of inhaled air are able to stimulate an anatomical response. The experienced perception is called odor. Odor assessment is a key point in food product processes to arrive at a quality product, likable and well accepted product too. Although the human olfactory system is still regarded as the most important and effective analytical instrument for odor evaluation, the demand for objective methods still exists. Many a time the comparison of subjective and objective methods of odor evaluation has been researched to arrive at a more confident, realistic picture of the odors and the compounds responsible for the sensing of odors. The analytical methods extension has led to the development of sensor-based machines, olfaction potentially imitating the biological system. The odor measurement is essential for odor regulation and control. Odor emission often consists of a complex mixture of many odorous compounds. Analysis of individual chemical compounds present is usually not practical. More often, complexity of odor involves psychological and physiological response, perceives the odor as pungent, ethereal, aromatic, pepperminty, musky, camphorous, putrid and so on, referring to the source. Therefore, the analytical methods can reflect chemical compounds responsible for these perceptions. As a result, odor sensory methods instead of instrumental methods are normally used to measure such odors. Broadly, odor measurement can be carried out by two methods. First is the sensory methods and second instrumental methods. Repeatability, reproducibility and accuracy have to be accomplished with any method. The sampling procedures, sample containers, olfactometer construction and operation, olfactometer and assessor interface, the odor testing room, methods of data processing, training of panel and consistency in their performance are of important concern for analysis. Now coming to sensory methods. Sensory measurements employ the human nose as the odor detector and human odor is directly experienced, which is related to properties. Sensory methods are available to monitor odor both from source emissions and in the ambient air. These two diverse circumstances require different approaches for measuring odor. The sensation of odor has four properties related to threshold and tolerance. They are odor concentration, odor intensity, odor quality and hedonic tone. Thus, sensory measurements techniques comprise of two categories. First is the quantitative measurements which couple the nose with some instrumentation, and second parametric measurements in which the nose is used without any other device. Instrumental sensory measurements employ the human nose in conjunction with the instrument called olfactometer. This dilutes the odor sample with odor-free air according to precise ratios to determine odor concentrations. Odor concentration is an odor's pervasiveness. To measure odor sensation, an odor is diluted to certain amount to reach a detection or recognition threshold. The detection threshold is the concentration of an odor in air when 50% of a population can distinguish between the odor sample and odor-free blank. The recognition threshold is the concentration of an odor in air in which 50% of a population can discern from an odorous sample and odor-free blank. The recognition threshold is usually a factor of 2 to 5 times higher than the detection threshold. To establish the odor concentration, an olfactometer is used which employs a group of panelists. The olfactometric measurement is affected by the olfactometer design, test procedure, differing sensitivity of observers and data quality. The materials used in olfactometer construction should not cause sample contamination. Low absorbency materials such as stainless steel, Teflon or glass are used and internal surface areas are minimized. The test procedure followed should have a proper choice of samples presentation to the panel, considering the olfactory adaptation in panelists. An ascending order of strong samples is preferred so that the panelists can detect higher diluter samples followed by lower dilution, that means to say stronger samples. Among the two standardized methods for the presentation of odor sample to the panel, single port used method is simpler than the forced choice multiple ports method. Sampling odor mixtures at different dilutions are presented to a group of selected panelists for sniffing and their responses are recorded. Generally, the first mixture presented to a group of an odor panel is diluted with a very large volume of air in order to be undetectable by the human nose. In subsequent sample presentation, the volume of diluent is decreased by a predetermined and constant factor. After having set the factor, it is possible to create a geometric progression of dilutions useful to describe the logarithmic relation between odor intensity and concentration. The process continues until each panelist positively detects an odor in the diluted mixture. At this stage, the panelist responses has reached the detection threshold for that odor. This threshold is expressed as threshold odor numbers and used to calculate the concentration of the odor in terms of European odor units, OU gram per meter cube. Panelists are qualified examiners used as sensors in olfactometric analysis, and their olfactive response, that is odor threshold, is the measured parameter for calculating odor concentrations. These panelists are selected according to standardized procedure to minimize the difference among their detection. The panelists should be the individuals with average olfactive sensitivity who constitute a representative sample of human population. The screening is usually performed using reference gases, and normally N-butanol odor threshold in a range of 20 to 80 ppb is the accepted limit, with 40 ppb plus 2.3. Panelists must be continuously screened and trained. Panelists with illness, cough, cold, allergy, etc. are eliminated. In order to present the odor sample to the panelists, samples must be collected using specialized sample bags. Sampling needs particular attention to avoid sample losses due to sorption on the container. The materials used for odor containers should be odorless, stable to physical and chemical reactions, and has low permeability. Stainless steel, polytetrafluoroethylene, that is PTFE, tetrafluoroethylene, hexafluoropropylene, copolymer Teflon, polyvinyl fluoride, polyterephthalic ester copolymer, naloxone and glass are considered suitable materials for odor sampling. The most accepted techniques for collecting odor samples is the lung technique. The sample bag is placed in a sealed drum and vacuum is placed on the drum, which fills the sample bag. As the bag expands, it draws the sample from the source into the bag. Critically, all components which touch the odor sample must be odor-free. The data quality is achieved when standard sampling techniques are followed. Representative sample of human population is used. Quality control protocols are followed during the conduct of experiments. The statistical analysis of the data, specifically ANOVA, which reflects the variance between group and within group, has to be adopted. The other instrumental based quantification of odor samples is studied using gas chromatography olfactometer, that is GCO. The GC olfactometer consists of a traditional GC where a split column distributes the eluate between a conventional detector such as flame ionization detector, that is FID, or a mass spectrometer, that is MS, and a sniffing port where a properly trained person or panel could detect the active odor species. The commercially available olfactory ports are glass or PTFE cones fitting the nose shape. The eluate is delivered through a dedicated transfer line which is heated to avoid the condensation of semi-volatile annihilates. In order to present the nasal mucous membrane dyeing, during long time analysis, auxiliary gas, that is humid air, is added to the eluate. The sensory responses are recorded in an olfactogram. The eluate splitting occurs, allowing the annihilates to reach both human and instrumental detectors simultaneously in order to compare the chromatogram with the olfactogram. The GCO technique is widely used for the evaluation of food aromas. The second approach for odor evaluation by parametric sensory measurements has the advantage of being quick to obtain at relatively low cost, as no particular equipment is required. The subjective measurement of parameters of odor perception by the well-trained panel. The parameters include odor character, odor intensity, and hedonic tone. Odor character, often called odor quality, is a nominal scale of measurement. Odors can be characterized using a reference vocabulary with a standard list of descriptor terms. Odor intensity is the relative strength of the odor above the recognition threshold, that is supra-threshold. Odor intensity is measured using several methods including descriptive category scales, magnitude estimation, and reference scales. There are several scales that usually employ 3 to 10 categories, and panelists must assess the intensity of the sample according to the specified scale. It is a verbal description of an odor sensation to which a numerical value is assigned. For example, the odor intensity is described as no odor 0, very weak 1, that is the odor threshold, weak 2, distinct 3, strong 4, very strong 5, intolerable 6. With a numerical value of 0 to 6, this test is followed in the laboratories and the odor intensity is done by a series of suitably trained panelists. Hedonic tone is the process of scaling odors on a scale ranging from extremely unpleasant via neutral up to extremely pleasant. The method is followed by serving the different dilution sample through an olfactometer to the panelists. If the panelist detects an odor, the hedonic odor tone of the perceived concentration must be evaluated according to the category scale. It is described as extremely unpleasant 4, very unpleasant 3, fairly unpleasant 2, unpleasant 1, neither pleasant or unpleasant 0, pleasant +1, fairly pleasant +2, very pleasant +3, and extremely pleasant +4. Moreover, it is important to note that perception of an odor may change from pleasant to unpleasant with increasing concentration, intensity, time, frequency, and previous experience with the specific odor. All these factors are determining the response. The overall sets of qualities are sometimes called as the fiddle factors, that is frequency, intensity, duration, offensiveness, and location. Now next, coming to instrumental methods. Though human perception of odor is desirable and regarded as most important and effective analytical instrument for odor evaluation, still there are problems in comparing different persons' experience in quantifying the odor. As a first step, olfactometer, called the dilution instrument, has been coupled with the sensory method to provide to the panel of human assessors. Later in 1988, electronic sensing technologies have undergone development for the technicalities as well as for commercial applications. Electronic sensing equipment, called e-nose or electronic nose, is capable of reproducing human senses using sensor arrays and pattern recognition systems. E-noses can detect and recognize odors and flavors. The stages of recognition process are similar to human olfaction and are performed for identification, including data storage and retrieval. Further, the development of a new olfaction system called electronic mucosa is based on advanced pattern recognition algorithms for space and time classification of odorants. Shea Harron et al., 2009. E-nose has been applied in the space shuttle to monitor air quality in the cabin. Willers et al., 2004. E-nose. The electronic nose consists of head space sampling, sensory array and pattern recognition modules to generate signal pattern that are used for characterizing odors in terms of perception as a global fingerprint. The major parts of e-nose are a sample delivery system, a detection system and a computing system. The sample delivery system enables the generation of the headspace, that is volatile compounds of a sample. The system then injects this headspace into the detection system. The sample delivery system is essential to guarantee constant operating conditions. The detecting system consists of a sensor set, a reactive component of the instrument. When volatile compounds come in contact, the sensors react through the interface transducer electrical signal. A specific response is recorded. A specific response is recorded by the electronic interface, transforming the signal into digital value. Recorded data are then computed based on statistical models. In most e-noses, each sensor is sensitive to all volatile molecules, but each in their specific way. However, in bio-e-nose receptor proteins, cloned from biological organisms, for example, humans respond to specific odor molecules through binding. The more commonly used sensors are metal oxide semiconductors devices. Besides this, the sensors are conducting polymers, polymer composites, quartz crystal resonators, and surface acoustic wave. Some devices combine multiple sensor types in a single device. In recent years, other types of e-noses have been developed that utilize mass spectrometry or ultra-gas chromatography as detection systems. Several studies concerning the use of nanomaterials as gas sensor materials have been reported. The computing system combines the responses of all the sensors which represent the input data. This instrument performs global fingerprint analysis and provides results that can be easily interpreted. E-nose results can be correlated to those obtained from sensory panel GC, GCMS results. The analysis with the e-nose as a first step needs to be trained with qualified samples so as to build a database of reference. Then, the instrument can recognize new sample by comparing volatile compounds fingerprint to those contained in the database. The limitation is the analysis of odors made up of multiple different molecules, which is normally detected by the device as different compounds. E-noses are mainly used in R&D labs and process and production departments. E-noses can be used in quality control mechanisms of conformity of raw materials, origin of vendor selection and detection of contamination spoilage, adulteration and monitoring of storage conditions. In the processing, it can be applied for managing raw material variability, comparison with the reference product to narrow down the batch variation, scaling up of process and monitoring the place safety. Considering the different techniques of odor evaluation, no one of the described techniques alone is able to exhaustive information about the odor emissions from different food systems as well from different kinds of human activities that cause olfactory picture. Therefore, a comparison or an integration of the olfactory methods with the methods of sensorial analysis makes the task complete to get a realistic picture of odors. Besides these, clinical test approach for detecting the odor identification ability of the panelists can be followed. However, the clinical testing can be time consuming and difficult to perform precisely. The major goal of sensory testing is to assess the chemosensory property. In this direction, simplified and standardized commercial kits are available. These can provide a reliable measure of olfactory ability. Tests of olfactory function can evaluate the threshold of odor detection, identification and quantification. These tests include butanol threshold test, the University of Pennsylvania smell identification test, that is UPSIT, and the sniffing stick test. The butanol threshold test involves a forced choice test using an aqueous concentration of butyl alcohol in one sniff bottle and water in the other. The person is asked to identify the bottle containing the odorant with each nostril tested separately. After each incorrect response, the concentration of butanol is increased by a factor of 3 until the person either achieves 5 correct responses or fails to correctly identify the bottle with 4% butanol. The detection threshold is recorded as the concentration at which the person correctly identifies the butanol on 5 consecutive trials. The UPSIT involves 40 encapsulated odors in a scratch and sniff format with 4 response alternatives accompanying each odor. The person takes the test alone with instructions to identify, if not, at least to guess the odor. The scores are compared against age, sex related norms and the results are analyzed. This test has excellent test-retest reliability. A variant of the UPSIT called Cross-Cultural Smell Identification Test, that is CCSIT, can be given in 5 minutes. This test gives a quick measure of olfactory function. The 12-item CCSIT has been developed using input on the familiarity of odors in several countries including China, Colombia, France, Germany, Italy, Japan, Russia and Sweden. The odorants chosen include banana, chocolate, cinnamon, gasoline, lemon, onion, paint thinner, pineapple, rose, soap, smoke and turpentine. The odor identification was most consistent. The test is an excellent alternative in clinical setting. It is rapid and reliable, but the disadvantage is that its brevity limits its sensitivity in detecting the subtle changes in olfactory function. The sniff stick method uses a series of reusable pen-like odor dispensing devices and tests odor threshold by a single staircase method. The overall evaluation of olfactory function is done by odor discrimination with forced choice among 3 of 16 different common odorants. For research studies, olfactory evoked response method is normally used. In conclusion, both subjective and objective standardized methods of analysis are described for odor evaluation. The sensory analysis with the nose as the detector with device proves to be simple to conduct but critical in analyzing the odor compounds. The combination of human with devices may be more fruitful with the single port olfactometer and qualified taste panelists. The instrumental analysis of odors is achieved using GC, GCMS, GCO, which is widely used for the evaluation of food aromas. However, olfactometry is time consuming and quite expensive. On the other hand, E-nose, which analyzes odors similar to human olfaction, remain as a promising method for odor evaluation. E-nose analysis present lower analysis costs and quick results. But the limitation is the large database required for identification. However, no single technique satisfies the totality of odor evaluation, and often a combination techniques are used for good end results. The clinical test can be adopted for quick analysis and for patients.